Hello, dear friends. I hope you all are well. I'm Nitin, and today I'd like to talk about the contribution of a Ukrainian microbiologist, ecologist, and soil scientist, Sergei Nikolaievich Winogradsky. He was born in 1st September 1856 in Kyiv, Ukraine. He'd completed Masters of Science in Botany in 1884. He further started his work at the University of Strasbourg under the supervision of Anton de Bary, who is considered as the founding father of plant pathology. De Bary suggested to Winogradsky that he could study Beggiatoa, a strange bacteria. Beggiatoa was strange because it didn't grow in the nutrient mixtures normally we use in labs. Yet, in sulfurous spring waters, it could develop into huge colonies, forming large mats. Winogradsky traveled to a number of different springs to personally collect samples of water, which he brought back to the lab. By 1887, the 31-year-old Winogradsky had discovered that Beggiatoa didn't obtain energy by any previously known method. Beggiatoa's energy came from a chemical reaction between hydrogen sulfide present in sulfurous waters and oxygen. Beggiatoa stored the sulfur it produced within its own cells, and if needed could react this sulfur with oxygen to produce even more energy. He returned to St. Petersburg for the period 1891 to 1905 and headed the division of general microbiology of the Institute of Experimental Medicine. In 1901, he was elected honorary member of the Moscow Society of Naturalists, and in 1902, corresponding member of the French Academy of Sciences. His discovery also includes the identification of the obligate anaerobe Clostridium pasturinum to be a free-living microbe capable of fixing atmospheric nitrogen and not living in the legume root nodules. Winogradsky invented the Winogradsky Column, which is still used today, to learn which communities of bacteria are present in a sample. It consists of a glass cylinder half filled with mud from a river or lake, topped with water. A number of ingredients such as sodium sulfate, calcium carbonate, and cellulose are added to provide source of nutrition for bacteria. Incubating this column in sunlight for months result in anaerobic, aerobic gradient as well as sulfide gradient. These two gradients promote the growth of different microorganisms such as Clostridium, Desulfovibrio, Chlorobium, Chromatium, Rhodomicrobium, and Beggiatoa, as well as many other species of bacteria, cyanobacteria, and algae. Within the column, scientists can control the number of nutrients and light available to the bacteria. Further, in 1922, he headed the division of agricultural bacteriology at the Pasteur Institute, Brie-Comte-Robert, France. During this period, he worked on iron bacteria, nitrifying bacteria, nitrogen fixation by azotobacter, cellulose-decomposing bacteria, and also devised culture methods for soil microorganisms. Winogradsky retired from active life in 1940 and died in Brie-Comte-Robert in 1953 at the age of 96 years. I have to say, he was a great man, and he'll always be remembered for his significant contribution to the field of microbiology. I'm really grateful to Archana ma'am for providing me the opportunity to speak on this topic. I'm so very thankful to the dear audience for listening and appreciating our previous videos. Please subscribe to the channel. Mentorship towards sure abundance. Stay tuned, keep learning, and be happy. Thank you and have a great day.